BTEC Applied Human Biology, Unit 1, A1, the cell cycle, mitosis and meiosis. Stem cells. So stem cells are unspecialised cells that retain the ability to become a wide variety of specialised cells. Specialised cells. As an organism develops, cells differentiate to form different types of cells. They develop to let it carry out a specific function. They include cells such as sperm, nerve and muscle cells. Totipotent. That means they can become totally anything. Stem cells with the potential to differentiate into any type of cell. Multipotent. Cells with limited potential to develop into many types of differentiated cells. Pluripotent. Cells that are capable of developing into most, but not all, of the body's cell types. Definitions. Haploid. An organism or cell having only one complete set of chromosomes. Diploid. An organism or cell having two sets of chromosomes. Homologous chromosomes. A pair of chromosomes that are the same size, same appearance and same genes. Somatic cells are body cells. Gametes. A mature haploid male or female germ cell that is able to unite with another of the opposite sex in sexual reproduction to form a zygote. Gene. A segment of DNA on a chromosome that codes for a particular polypeptide. Locus. Location of a gene on a chromosome. Cell cycle. A series of events that cells go through as they grow and divide. G1 phase. Stage of interphase in which cells grow and perform its normal functions. Releases energy and protein synthesis occurs. S phase. The synthesis phase of the cell cycle, the portion of interphase during which DNA is replicated. G2 phase, stage of interphase in which cell duplicates into its organelles. Cell cycle checkpoints, mechanisms that monitor the preparedness of a eukaryotic cell to advance through the various cell cycle stages. Alleles, different forms of the same gene. Sister chromatids are identical copies of a chromosome. Full sets of these are created during S subphase of interphase. Centromere is the area in which chromatids of a chromosome are attached. A bivalent is a pair of homologous chromosomes. Mitosis is cell division in which the nucleus divides into nuclei containing the same number of chromosomes. Two genetically identical daughter cells are produced. Mitosis uses: growth, repair, asexual reproduction. Stages of mitosis: PMAT. Prophase, metaphase, anaphase and telophase. Prophase is where the chromosomes condense and become visible and the nuclear envelope dissolves. Metaphase is where the chromosomes line up at the middle or the equator of the cell and spindle fibres start to form. Anaphase is where the sister chromatids separate and move to opposite ends of the cell. Telophase is where chromosomes unwind and the nucleus starts to reform. And cytokinesis is division of the cytoplasm. Meiosis now. So meiosis is nuclear division that produces gametes. 4 genetically different haploid daughter cells are produced. There are 2 nuclear divisions. Meiosis occurs in the testes and ovaries, and variation is achieved by crossing over and independent assortment. Fertilisation is when 2 haploid gametes fuse and the diploid chromosome number is restored. This ensures that in sexual reproduction, fusion of the haploid gametes restores the original chromosome number, the diploid, and that chromosome numbers are kept constant. Stages of meiosis. Meiosis 1 is where the homologous chromosomes separate. Meiosis 2, the sister chromatids separate. Crossing over is a process in which homologous chromosomes exchange portions of their chromatids during meiosis. So you then need to know the individual stages in meiosis 1 and the individual stages in meiosis 2. So it's PMAT twice. So prophase 1, crossing over, chromosomes become visible, nuclear envelopes breaks down, crossing over occurs. Independent assortment. Independent segregation of genes during the formation of gametes. They can line up randomly in any orientation at the equator during metaphase. Metaphase 1, paired homologous chromosomes line up across the centre of the cell. Anaphase 1, homologous chromosomes separate. Telophase 1, A nuclear envelope forms around each cluster of chromosomes. Prophase 2. A new spindle forms around the chromosomes. Metaphase 2. Centromeres of chromosomes line up randomly at the equator of each cell. Anaphase 2. Sister chromatids separate. Telophase 2. A nuclear membrane forms around the chromosomes in each of the 4 new cells. Mitosis v meiosis. Mitosis has 1 nuclear division. The chromosome number remains the same. It's for growth and repair, produces 2 daughter cells. Daughter cells are genetically identical to each other in the parent cell, and there's no crossing over or independent segregation. In meiosis, there are 2 nuclear divisions. The chromosome number is halved. It's for the production of gametes. It produces 4 daughter cells. Daughter cells are genetically different to each other and to the parent cell. Crossing over and independent segregation occurs in meiosis 1.